morning. Welcome again for this tips and tricks for practical exams. And if you have seen the breakup of your practical exam, you know that it has a viva component which is for 20 marks. And in 20 marks, you'll be at least asked 10 questions. This is, I believe. So I try to tell you few questions which I will like to ask in the viva. And I start because in the practical you will be asked the questions which are related to your practical. So let's start with the questions, and I have written down few questions, and let's do them one by one. First question, which can very easily asked is, what is the chromosome number in onion? Why this question is asked very often is because in your files, in your drawings, you will show them the chromosome number of 2, 3, 4, 5, which is convenient for you to draw. So please, you should know this, that the chromosome number in onion is 2n is equal to 16. And then you have learned this in your practical. So if you are asked, you can answer it's 2n is equal to 16. The other question, then you will have questions related to staining, that why do you use acetocarmine for staining in mitosis and meiosis? And you can always answer this, that acetocarmine is a non-specific nuclear strain. So it will strain the nuclear region and it will show you the chromosomal region easily in that. So you use acetocarmine for that. And after that, you may be asked, can you name any specific strain or any alternative for this one? Then you got to say here is that you have fulgin, which is used for specifically staining the DNA, and for sometimes it's used for quantitative estimation also. And then this question may be dragged to different forms of staining, forms like any alternative or how does acetocarmine work, and then you have to tell that it is a non-specific nuclear strain. Then from where acetocarmine is obtained, you may be asked about that, and you have to answer that it is an insect, Coccus cacti. And then you may also be asked, why do you heat while staining with acetocarmine? And here you have to say that we use 1% acetocarmine in acetic acid, which is 45%. And we stain this, and to fasten the staining, we heat this, meaning in the squash of onion root, you heat it a bit so that the staining is fastened and you are able to get result quickly in the class. The process is slower without heating. So you have to explain why you are heating also. And then you observe the slides under the microscope and look for the stages. And then the other question which may come, what is the difference between acetocarmine and fulgin staining? That you know now already, that fulgin is specific for DNA and may be used for quantitative estimation of DNA also, but it's relatively non-specific when we stain with acetocarmine. Then suddenly you may be asked, because sometimes they divert, who coined the term mitosis? You answer Walter Flemings maybe. And then about colchicine also, it's a very common and a favorite question. What is colchicine? What for it is used, and where do you obtain this from? So colchicine, you need to answer is that it's a mitotic prisoner, and it will arrest the spindle fiber formation, and it is used to induce polyploidy, and you obtain this from the bulb of a plant, that is Colchicum autumnale. It belongs to the family Liliaceae, and common name of this is what? Meadow saffron. It's a very, very common question in the practicals also it is asked, and in the theory also it comes very often, and it strikes you here with the staining that you may be asked for this. You might also be asked which stain you want another stain to specifically bind to DNA. Then you have to answer here the fulgin again. Then sometimes you may also be asked, because you have karyotype in your syllabus, ideogram karyotype, which stain do you specifically use to stain chromosomes? And then again you have to say Giemsa stain. And Giemsa stain, you have to explain its working, because it differentially binds to heterochromatin and euchromatin. That is how it gives bands. So an alternative to fulgin, maybe in studying the DNA, you may be asked about Giemsa strain also, drag to that question after this. Then you may also be asked why there are no stages very often when you start studying mitosis. So you got to explain here that these are the meristematic cells which rapidly divide. That is why you use onion root tips. And these tips also sometimes they have a cell cycle and a duration and a timing where they divide very rapidly. So to get stages, you must have the material which has been fixed at a right time so that it is dividing rapidly. Otherwise, then you have to struggle to get the stages. One very common question, again coming to the stages and mitosis, meiosis, is that what is synchrony? And synchrony, when all the cells in an organ or in a maybe anther, they divide at the same time. This is synchrony, and this you observe in meiosis and not in mitosis, because in meiosis, in the pollens, you will see all the cells are dividing at the same time. So when you observe, you make slide, you see that several cells or pollens, they are dividing at the pollen mother cells, giving you pollen, and lots of pollens are coming out simultaneously. Then another very common question coming up is how will you distinguish between metaphase 1 and metaphase of mitosis? So you must have the answer ready for this. But a quick answer to this one again is that in mitosis, what happens? The centromere divides and you get the chromosome duplication. And in meiosis, what happens? And in the bivalents, the homologous chromosomes, they separate. And you have to prepare this answer very well. It is very likely to be answered to be asked in the viva. Then probably, because I think it's again getting a very longer video, I'll just wind up with this last question. What is the best time to fix the material? If I ask you to fix a material for preparation in the laboratory, what is the best time you have learned to be fixed for onion root tips? So that you have already known that every cell has a cycle, and the onion root tips are known to have a cycle of 24-hour duration. So they divide once in 24 hours, and this division takes place about 2 hours after sunrise. So nowadays, when the sunrise is taking place at 5, so the best time to fix the material would be at 7 a.m. in the morning. So if you are not given a proper material which is fixed up around this time, it is very likely that you will get much less stages. So you need to answer if you don't get stages, and if you answer this question, sometimes the examiner would think that you know the things and it's none of your fault, so you should get the marks. So these 15 quick questions I could think of, but I think this is not all, because I have to have questions from DNA extraction and DNA staining, and then the ratios also. So I'd like to see you again. I'll meet you again in a while. Whenever I get time, I'll record them and send it to you. So okay, bye for now, and I look forward to see you again. Thank you.